We're very excited today to have with us is Rene van Tienhoven. He is a PhD at the Department of Cell and Chemical Biology at Leiden University Medical Center, and his talk today is Genetic Beta Cell Protection by Nature. A little bit about him. He studied biomedical sciences at the University of Groningen. He conducted his PhD research in the lab of Bart Rope and City of Hope in Los Angeles, focusing on the expression of neoantigens in human islets and revisiting the role of the insulin gene in type 1 diabetes. He successfully defended his thesis in January of this year, and currently he continues his research on type 1 diabetes as a postdoctoral fellow at Leiden University Medical Center, where he aims to understand how beta cells provoke the immune system and ultimately to contribute to finding a cure. We will have everyone dropping questions into the chat as the talk progresses, and we'll also take a Q&A at the end of the presentation, and I'm also going to drop a couple of his newest papers in the chat for your reference. And thanks so much, Rene, for joining us. This is a very
Yeah.
exciting paper, and there's a lot of buzz around it. So we really are look forward to your presentation.
Yeah, thanks for having me and thanks for the introduction. It's nice.
Sure.
Yeah, indeed. So we found a way how to protect beta cells basically with a gene, and it's kind of in a natural way. So that's what something that we like about it. Type 1 diabetes is always seen as an autoimmune disease in which the immune system here attacks the innocent beta cells. But we and others have shown that the beta cell also plays a part in this disease. So the beta cell provokes the immune system to attack. Certain genes can increase the risk of type 1 diabetes. HLA has the biggest odds ratio to do this, but insulin is the runner-up. And the insulin gene can be either protective or susceptible regarding regarding type 1 diabetes risk. So this gene comes in two flavors. The protective one you see here on top, it has a long VNTR and these protective SNPs that are in tight linkage disequilibrium. And if you have this variant, then the odds ratio of getting diabetes is only 0.5. So you're basically 50% protective. Now, if you have the susceptible variant, the risk is 2 times higher compared to normal. And then you have the short VNTR with susceptible SNPs. Now, this protective mechanism has always been explained by the VNTR, but it's interesting to say that they typed the VNTR by using these tagging SNPs in the back. So we are having a hard time saying what the causal SNP exactly is. But anyway, it has been explained by the VNTR, and this was by the immunological perspective, as we call it. So if you have the protective variant here on the left, then they found that you have insulin expression in the thymus, whereas if you have the short VNTR, then you do not have insulin expression in the thymus. Now, if we take a look into the thymus, then and you have insulin expression here, then your T cells that respond to insulin will basically go into apoptosis, and that's what we call central tolerance. Now, if you do not have insulin expression, so with the susceptible variant, then you have these autoreactive T cells that can respond to insulin and could attack your beta cells. Something else that they found was that there is lower expression of insulin in the pancreas, so in the beta cells, if you have the protective insulin variant, but they never really explained why.
Quick question from the chat, Rene. Are the odds ratios of 0.5 and 2 for homozygous expression?
No, that's for heterozygous. So if you have only one variant, you're already protected, and that protection is dominant. So something else that we were struggling with is that this central tolerance cannot really explain everything that we see in the literature. So what the Renovic-Bellows showed in 2005 is that if they see T cells that respond to insulin, then these T cells make IL-10 if you have the protective insulin variant, but they do not make IL-10 if you have the susceptible insulin variant. And a couple years ago, in this paper of Asfalk, they showed that with the protective insulin variant, patients have more Treg-like T cells and a lot less effector-like T cells that respond to insulin. But this is swapped around if you have the susceptible SNP. But this could not really be explained by central tolerance, but can be explained more by peripheral tolerance of regulation. Now, in our own data, we also found something like this, where when we were looking at T cells that respond to insulin drip, we found that only patients with the susceptible insulin gene can respond to drip, but patients with the protective insulin gene did not respond to drip. You see that there. And we do see this change, but central tolerance against a neoantigen is not very likely because it's presumably new, so not expressed in the thymus. So we wanted to see what's going on in the periphery, so what's happening with the beta cells. Also because insulin is, of course, expressed in the beta cells. Now, if a beta cell is happy and healthy, it makes insulin, our favorite hormone, I think. But when beta cells get stressed, they can start making mistakes. One of these mistakes was insulin drip, what I was just talking about. Um, and this happens if the translation is started at another AUG. So the ribosome is basically making a mistake and making a different protein from the insulin mRNA. And then we get this neoantigen insulin drip, which can activate T cells. There's also possible to do alternative splicing. So that means that the introns and extrons are spliced together differently. And then we get insulin splice. That's a protein that looks like insulin for the first bit, but then looks like drip for the later half. And there's also other things that can go on, like deamidation of proteins that can lead to immunogenicity and provocation of the immune system. So what we would like to do is make these stressed beta cells happy and healthy again. But to do that, we first need to see what is going on during this stress. So here we have a normal endoplasmic reticulum. And if we zoom into that, we see that during normal conditions, the proteins inside are nicely folded into their structure. And that this protein BIP is bound to these ER stress sensors, and that basically inhibits them. But now, if there is stress, that means that there's unfolded proteins in the ER, and then this BIP protein can, it would rather bind these unfolded proteins than the stress sensors, and that makes, therefore the stress sensors can activate. And then what that does is it turns on the unfolded protein response, and that is a response to get rid of the unfolded proteins and make the cell happy again. And how IRE1 does that is by splicing XPP1 into XPP1 spliced. And XPP1 spliced is a transcription factor that goes to the nucleus and improves the protein folding. Something else that IRE1 alpha does is it can break down other mRNAs, and that it does that to reduce the translation. So it, on the one end, it improves the protein folding. On the other end, it makes sure that there's not too many proteins being made at that time. And this process is called regulated IRE1 dependent decay of mRNA. So we like to say that it gets rid of mRNA. And IRE1 doesn't cleave just all mRNAs. It only cleaves mRNAs that have this specific motive. And now we were wondering, could one of these other mRNAs be the insulin mRNA? Now, this is just a little refresher of what the insulin mRNA looks like. So here we see the canonical AUG of blue in blue. I hope you can see my pointer. And that translates just to the normal insulin sequence, right? But we recently discovered that during stress, the ribosomes can make a mistake and start translation at the drip AUG, creating a neoantigen. And you can see that it's in a different frame of insulin, and it makes a completely different protein. Then when we come to the stop codon of insulin, because it's in a different frame, insulin drip will just keep going until it comes by these SNPs in the 3' UTR that we were talking about earlier. And then we see that it can translate these SNPs into polymorphic proteins. Now, something else that we noticed was that exactly where this SNP is that this is the same motif that IRE1 alpha can recognize. And the interesting thing is that it's only there if you have the protective insulin variant. Now, this motif is not the only prerequisite that IRE1 alpha needs. IRE1 alpha also needs this hairpin in order to recognize the mRNA. So we folded these, the protective insulin mRNA and the susceptible insulin mRNA using a computer program. And indeed, we found that this motif is present in the protective insulin mRNA, and that IRE1 alpha can recognize it. And our hypothesis is then that the protective insulin mRNA can be broken down if the cells are stressed. And that leads to a reduction of the translational stress. And it also gets rid of the drip neoantigen because it's also made from the same insulin mRNA. And that leads to less immunogenicity and ultimately protection from type 1 diabetes. Now, if you have the susceptible insulin variant, there is no hairpin and no motif for IRE1 alpha, so it cannot break it down. And then your insulin mRNA stays intact, the translational stress remains, and drip can still be produced from this insulin mRNA, making your beta cells more immunogenic and leading to susceptibility of type 1 diabetes. But does this actually happen? Was the question. How we try to answer this is by isolating mRNA from donor islets and then adding pre-activated IRE1 alpha into the mix and then looking at the decay of insulin mRNA. And as you can see, it does indeed happen. So insulin mRNA can be broken down. If you have one protective variant in dark blue, then the breakdown is faster. And if you are double protected, then we see that in 20 minutes, all of the insulin mRNA is already gone. So it really seems that the insulin breakdown is possible. And we see that in this case with purified mRNA and a purified protein. So next, we also wanted to see if this happens in the cell itself as well. And for this, we used a little trick of nature with the firefly. So what we did is we did a dual luciferase assay with the vanilla luciferase and the firefly luciferase. And we cloned the susceptible insulin UTR or the protective insulin UTR behind the vanilla luciferase sequence. And then we checked whether the vanilla luciferase activity was different between the two. And firefly luciferase here is sort of an internal control that is transfected together with every factor. And when we did that with increasing ER stress on the x-axis, you see that if you have the protective, if we transfected the protective SNP, sorry, then the knockdown is 3 times higher. So there's a lot more breakdown with the protective version compared to the susceptible version. So next, we also wanted to see if this was specific for IRE1 alpha, and that's why we use this specific inhibitor. And then you see that if IRE1 alpha is inhibited, that the difference between the two SNPs is no longer significant. So the next step was to see if there's also an effect on immunogenicity. And to test that, we wanted to see the difference between stress translation and normal translation So what we did is we added this GFP tag in the reading frame of DRIP, so that whenever there's a ribosome that's starting translation in DRIP, we would see GFP, but in normal conditions, we would just see, well, nothing basically. And when we transfected these cells and again added stress, we saw that there's a lot more DRIP positive cells if we do this with the susceptible variant compared to the protective variant. And also the amount of DRIP that was on these cells is higher with the susceptible variant. So that basically means that the cells are less immunogenic because they have less DRIP neoantigen when they have the protective insulin gene. So next, now we wanted to see the effect on real islets and patients. And we did that by looking at two different cohorts. So one of the cohorts was from HPAP, and I would like to thank HPAP also for making all that data available. The other cohort was from our own hospital, City of Hope, where we isolated islets and tested the function. Next, we also looked at complications of in patients themselves. But I will start with the HPAP data, and this is something that John Cadiz and Dennis really helped us with this analysis. So we selected 8 protective donors. That were all the protective donors that we found in the program, and then we matched those with 8 susceptible donors, looking at BMI and looking at age. And then we made a UMAP of all the cell types. And then we can see that here we have our beta cells. And these beta cells, of course, make insulin. And they have MAF-A, so they're really beta cells. So now, since we distilled these beta cells out of the equation, we could look at the insulin mRNA levels with either SNP. And we made a distinction between nascent insulin mRNA and mature insulin mRNA. And nascent insulin mRNA is basically pre-mRNA that has not yet been spliced. And that says something about the promoter activity, because that was the prevailing hypothesis. And what we see is that there is actually no difference between the protective insulin, between nascent insulin mRNA, looking at the protective or the susceptible. So it seems that the promoter of insulin does not really change with either SNP. Now, when we're looking at mature insulin mRNA, we can look at the mRNA that's left over, but to our surprise, we also did not see a difference between the protective and susceptible insulin variant. So after some thinking and reflecting, we figured out to add beta cell stress into the equation. And this way we see that there is more insulin mRNA with lower stress, but then when the beta cells get stressed, they have less and less insulin mRNA. But importantly, this only happened in the blue protective version, and this effect was absent in the susceptible beta cells. So this again shows that during stress, the insulin mRNA can be broken down if it is from the protective variant. Then we wanted to know if this insulin genotype also affects other genes. So we did a differential gene expression analysis. And interestingly, we only found 2 genes that were different between these beta cells. One of them was a link RNA that we don't know too much about. But the other one was very interesting. That's TGM2, and it's higher expressed in susceptible beta cells. And it's interesting because TGM2 codes for tissue transglutaminase 2, and that is known to create neoantigens by deamidation. And how that works is as followed. So here we see a native islet peptide, right? And it kind of fits in the HLA pocket, but not very good. But then when tissue transglutaminase deamidates these 2 amino acids, it fits perfectly like the lock in a key. So this is another way how the protective insulin gene can lower immunogenicity in the beta cell. So not just by only looking at drip, also other neoantigens could be, will be lower expressed in protected beta cells. Next, we will look at the City of Hope cohort, and in that cohort, we compared the, the function of the beta cells. Here we're looking at the OCR during basal respiration, and then if we challenge the beta cells with high glucose, we see that the mitochondria use up a lot of oxygen, especially when the beta cells are from the protective genotype. So in other words, islets with a protective genotype are more ready to respond to a glucose challenge than the susceptible islets. And here you see the OCR fold change that is so from basal conditions to maximal respiration conditions. And interestingly, is this point right here that was the one donor that was homozygous protective. And that had the highest OCR fold change of all the donors. So what this means is that the mitochondria of these beta cells, of the protective beta cells, are better and that they can make more ATP. So the next question was, can they also secrete more insulin? And the answer is yes, like you see here. Both in low and high glucose conditions, the protective islets make more insulin compared to the susceptible ones. And again, the homozygous protective donor is here in light blue, making most insulin of all the islets. Now, when these islets were transplanted into mice, we see that the protective islets reverse type 1 diabetes, or no, not type 1 diabetes, but they reverse diabetes in these mice twice as fast compared to the susceptible islets. And that's something that you can see here again. So all the mice reverse diabetes within 14 days. But with the susceptible islets, it took almost 28 days for all the islets to, for all the mice to reverse their diabetes. So it seems like all these, with these tests that we really show that islets with the protective genotype function a lot better compared to the susceptible ones. Now, despite all these benefits, some people with the protective insulin gene still get type 1 diabetes, but they do seem to have a milder disease progression. So here on the top, we see that they have less autoantibodies against insulin compared to the susceptible genotype. And on the bottom panel here, we see that they have a lot more stimulated C-peptide compared to the susceptible one. So that seems that the beta cells that are left over make more insulin. And in these patients, this also led to better glycemic control and lower HbA1c levels. So for this reason, we wanted to look at the complication incidence in these two groups. And for that, we used data from the DCCT EDIC trial. And we saw that there was no complications at all if you have the protective genotype compared to the susceptible genotype. So patients with type 1 diabetes that have this protective genotype are completely protected from complications. And I do need to say that this was only the case when they were also treated with intensive insulin gene treatment. But that is common practice right now. So the take-home message is that if you have the protective insulin gene variant in your beta cell, then you have a lower risk for type 1 diabetes. You can break down insulin mRNA. That leads to less ER stress and a way to get rid of drip, so lower immunogenicity. And as a bonus, we also see that these beta cells function better and secrete more insulin. Now, these are the implications for the future and the future directions that we want to go into. So we discovered a new causal insulin variation with a novel mechanism of protective action. We see that beta cells that carry this protective variant are less stressed, less immunogenic, and more vital and functional. When patients get type 1 diabetes, despite having this protective insulin variant, they show less autoimmunity and they do not respond to insulin gene products. They do not develop complications with eyes or to kidneys. And it seems that they represent an endotype of type 1 diabetes. Now, in the future, we would like to test whether insulin SNP correlates with disease progression or therapeutic intervention, and whether the insulin SNP defines the outcome of islet stem cell donors. And we would like to develop iPSC-derived islets with the benefits of the protective insulin variant. So lastly, I would like to thank our team from Leiden and our old coworkers from City of Hope, especially John Cadiz and John Williams, and the islet isolation team for all their help during these projects.
Great, thank you very much. I'm going to open it up to questions. It was an excellent presentation.
Was a little quick.
No, it was perfect. Anyone who has a question, you can unmute yourself, raise your hand, or drop something in the chat. I had a question, would you hypothesize that the insulin messenger RNA in the protecteds is broken down sort of like contextually? I mean, is it just sort of, is it the viral insult is, causing more of it to get produced, right? As the viral machinery takes over and then it's getting broken down. But what other things can kind of set the stage for it to be broken down more easily in those that are protected?
Yeah, what we looked at was not necessarily the viral insult, although that can definitely there's definitely a footprint of a virus that could have been there, or it could be
Except no
also
virus.
of RNA that
Wait,
could lead to this
someone
interferon gamma.
has broken into the call.
Yeah, but what we were looking at was more the ER stress, so endoplasmatic reticulum stress, and that leads to the activation of this enzyme IRE1 alpha.
Yeah, but I
I'm
mean,
not sure if it's
the
also involved
endoplasmic
in the.
ER stress happens in viral insult too, right?
I guess so, because a lot of viral particles need to be made.
Mm-hmm. I
I'm
don't
just
know.
not sure if that happens in beta cells. That's
Mhm. Well, like if you could imagine, and anyone can weigh in on this too, but like if Coxsackie virus is infecting, um, someone who has this protective, um, you know, phenotype, then they are, possibly able to kind of just not have the buildup of the insulin messenger RNA or the neoepitopes of the drips. And so under the stress of Coxsackie, they would come out unscathed, where there's somebody who doesn't have that could potentially get type 1.
Yeah.
Monica, let's not have this beautiful discovery hijacked by virus. There is no virus in beta cells, so
Oh, there's none
it's
in
not.
there's none in beta cells at all? Okay. Okay. Let's see. Has this validation been evaluated in responders and non-responders in previous immunologic therapy trials?
No, that is something that we want to do right now. That we are trying to figure that out to see what trials did actually yeah, do a genetic analysis, like to see what SNPs they have. I mean, not every trial probably has that. But that's something that we're trying to figure out. Like, does having the protective SNP give you a different outcome to certain trials than having the susceptible SNP? Because you could
Yeah.
imagine that with the protective SNP, you might not need to suppress the immune system um, because the immune system doesn't really attack as much, for example.
Yeah, more personalized medicine and addressing the endotypes.
Yeah, that's where we want to go. Yeah.
Here's another one. Did you check the agrosomes in the ER under stress to indeed see the misfolding proteins?
No, we did not look into the agrosomes. No,
Okay,
we
so
only we used the ER stress as a model of turning on the enzyme,
yeah,
and then we looked what the effect was with either the protective or the susceptible insulin mRNA variant. But we did not look into the cause of the ER stress in the beta cell.
more to be done.
But Rene, we did see the drip in the stressed beta cells, right? I mean, maybe not. We did not look whether they were in the agrosomes, but you could detect it in stressed beta
Yeah.
cells, right?
Yes, that's true. Yeah. So we saw a stress protein in stressed beta cells indeed, and
Hmm.
more of it in the susceptible variant than with the protective variant.
Mhm.
Yeah.
Matthias van Hereth.
Yeah, I was wondering, Rene, whether you or also Bart, whether you feel that this could be part of a bigger picture that's going on early on in type 1 diabetes. Because when you look at what we see, right, we have, at least people have alleged pro-insulin processing defects. We have this accumulation of MHC class 1 that's probably independent of any immune infiltration early on. And then I called it the constipated beta cell because it was nebulous to me what actually happens with the beta cell. Right? You say it's an endotype, but that endotype might just superimpose on or even work together with a bigger problem where the beta cell just gets lazy. And we always say it's ER stress, but ER stress, we have said this for 20 years. There has to be something more specific because I would argue also in type 2, we have ER stress. But type 2, except for some getting also autoimmunity, in general, 90% patients of type 2 have maybe ER stress, but they don't get this autoimmunity. So my question is, pathway-wise, like concretely in pathways, if you could have a guess, because that's not my expertise, but what could be going wrong there? Why are the beta cells getting lazy, not processing the insulin and degrading it properly? Why they're not cleaning house anymore?
Yeah, there
Why
must
they're
be a
getting
lot of things
depressed and
going
not
on.
doing anything anymore?
Yeah, it's interesting that you say that the beta cell is full because when we try to explain this finding to journalists, we said that the protective insulin variant basically gives it a valve to blow off steam,
Exactly.
to not be so full of mRNA and not be
I think
full.
Bart, we should write a paper about constipated beta cells. This would be a fun paper to write.
You make a wonderful point as always, Matthias, and what I like about this story is not so much how you get diabetes, but how you not get diabetes. So if you
Agree.
have a way to relieve stress, so the opposite direction, that's a way to avoid that you even get into trouble. Now, how you get it, that's a different story, and I don't think that Rene has shown any data on that part, but yeah, I very much like the constipated beta cell because it all has to do with unfolded protein responses and so forth, right? And but mostly how you deal with it. So anybody can get it, but many of us can deal with it. And the other variable in the equation is, of course, the immune risk. So the number one risk gene is HLA. So you could have ER stress, but not having the HLA that makes your immune system too trigger happy, and then you still would avoid destruction. So you're right. I think this is actually getting to the heart of the problem.
Maybe one could even hypothesize, and I mean, of course, it's difficult to get with that at the root cause. But I remember some years ago, I was discussing this with Kevin Harold, and we were saying, how would that be if like these pro-insulin specific cytotoxic T cells that we have all been observing, maybe these are our natural regulatory cells that come out there and they regulate the scene, and then other cells, it could be a few ones, right? Because it doesn't take so many. And you would, of course, say, well, Matt is, of course, the modified antigens. But we don't know that for a fact. But it could, of course, be that at the end of the day, it works like this, right? You have natural antigens that tolerize, and then you have all of a sudden this constipation going on, and that natural tolerization breaks down, and then it just takes a few modified antigens that come up and that trigger the immune system. Sorry, I don't want to battle things as Alicia has a comment. Sorry.
I was going to say, Alicia, would you like to weigh in here? She's been waiting.
Can I just make a quick response to this before you go ahead, at least because I think you're absolutely right. And remember how darn hard it has always been, Matthias, to find immune responses against native proteins, right? You really have to be a French chef or an Italian chef to be able to get those responses measured, whereas it's so simple to find against a neoantigen. So I think you're right on the spot there. Sorry, Elise, go ahead.
No, I just wanted to make a comment. I really enjoyed the presentation, and I was excited when I saw your paper initially. And my thought, I mean, I saw it in your conclusions that I think it would be really cool to apply this to transplantation, especially iPSCs, and see what happens. Because, I mean, my theory about transplantation is, like if there was no stress, there would be no rejection because such a small amount of antigen that is there compared to a whole organ transplant. So I would be really excited to see what happens. Congrats.
Yeah, there's always a
Beautiful
lot of
work.
stress going on, right? When they transplant islets, and we are hoping that if we give stem cells or islet donors that we select them with the protective SNP, that then they will just get the little advantage that they need and be able to
But I mean, Alicia's point is good. With the increase in technology, maybe even microencapsulation, not occlusive, but just holding them there, maybe with some windows even, right? You might be able, if they're stem cell-derived beta cells, islets, I mean, they're uniform in size, you can clean them up. That stress prior to transplant might become even further reduced. And there is something to be said, like you said, Alicia, if we get over the first hump and not all hell breaks loose and we have all these antigenic release, if we can at least avoid altogether these first priming events, that would probably be really, really good, right? Because it sets the stage, towards a really downhill trajectory if you have these first priming events. Sorry, Bart.
No, you're absolutely right. I mean, first impressions matter. And Alicia, I also believe that the immune system is not interested in happy tissue. So if you can give the tissue a kickstart, having happy islets, I mean, how good can it be? So I completely follow your reasoning. Thank you for bringing this in.
Well, we had Ralph Paus was talking at DRI yesterday, and in the hair follicle is same story, in alopecia. It's like staggeringly similar. It's like a happy hair follicle doesn't attract any immune system.
But only you see it in RA, with the smoking and the situations. I mean, I think this is a common theme that's emerging.
Yes.
Yeah, going back to the immune system's just doing its job. Ben Geipmans.
Exactly.
In your mouse rescue experiments, seemingly susceptible SNP islets are divided into two groups. One similar rescue to protective, the other takes longer time to rescue. Did you correlate these patients' complications? Might the two different group comparisons of susceptible SNPs be food for thought for the next protective variant mechanism?
Okay, let's see. So they were not patients. They were non-diseased donors. They were not healthy, but they were. Let's see,
It's a bunch of questions.
Yeah, but this is along the line of what Alicia was asking, right? So indeed, when you do human islets, transplant them in immune incompetent mice, you already see that it's much easier. And so that's even without an adaptive immune response, it's much easier to reverse diabetes. So this is something we definitely would like to study.
I guess, yeah, so I guess that's food for thought for the next protective variant mechanism. I wonder if you guys have thought much about Yuval Doer's paper in Cell Metabolism for January 2024. You know, diabetes, type 1 diabetes might be driven by non-viral endogenous double-stranded RNA. And then when they disrupted that RNA editing enzyme, ADAR, in the betas, they triggered this massive interferon response. There's this whole glycolysis, upregulation of calcium, and then increase of interferon response. Like, he did a lot of that work in mice and in type 2, but, you know, could there be something, you know, with this mRNA realm that dovetails into what you're talking about?
Yeah, I definitely think that this is one of the ways how we can get that interferon signature in a beta cell without even a virus being present.
Mm-hmm.
I'm not sure if IRE1 alpha, if that's what you're asking, if that can also cleave these these viral repeats that
But what it has in common, Monica, it's a great question again, is that it's about viral sensing, right? And
Yes.
IRE1 alpha can do it, but you know, double-stranded RNA, that is the ADAR1 story of Yuval, which I think is brilliant. And René's new mentor, Arnaud Zaldoumbide, he is actually very much into this and has shown the same thing, that you can have sterile inflammation and recapitulate anything you see, what people otherwise call infected. But it does kick off ER stress. It does kick off interferon alpha, upregulation of class 1, and the whole, you know, what Matthias used to call fertile fields for things to go wrong. I think that Yuval is on top of this, as is Arnaud.
Yeah. It just seems this whole, you know, your group, their group, everyone is sort of circling this RNA response or sensing mechanisms.
Yeah.
I think it's a new way of looking at the problem, and it's seemingly important now, you know.
And what it has in common, which I think is extremely interesting, that it's actually the beta cell where it all happens, right? And I honestly believe, I'm not sure whether you said it, Monica, or somebody else, but immune system is just doing its job, right? I mean, it don't blame it. It's actually really trying to fix it. And you're also right, Monica, saying that infection does create neoantigens and drips, as does cancer. And this is how the immune system has been educated to surveil and act upon it. And that's what's happening. So if you really want to do something, immunotherapy will be insufficient, and you need to have beta cell therapy. And one of our key targets is, you know, liraglutide and things like that, just making beta cells happy again, and then create a perfect storm for the immune system to get back in check.
Well,
I mean, we should maybe, Bart, we should write about some of this. Of course, when it comes to GLP-1 class, I have conflict of interest. And you
really?
can say maybe I'm biased. But look, I have been fighting this issue, to be honest, that it can have causative changes, also based on our organoid systems that we did together with Jonna and Phil. Because right now, these GLP-1s are classified as adjunctive therapies. And it's driving me nuts, because adjunctive therapies is literally looking like you're already doing well, but you put a face cream on so your face shines a little bit better. It's just like there's this notion that you're adding something to something that's already really good. But there could be causative therapies, right? That's the notion one would have to get through to the community, because I couldn't agree with you more, but the way we've seen the pathology, the data coming out in the last 10 years in particular, this is at least 50% a disease of the beta cell. Yeah. And it would explain why after immune therapies, you always see this initial blip of hopefulness, and then that goes away with most of the therapies, right? Even, you know the Voltarelli trial better than any of us, but even if you do this non-myeloablative bone marrow transplant, the benefit is limited, right? So that really intrinsically argues that there's a beta cell driver we are not fixing. right?
But Matthias, doesn't that automatically support the adjunctive therapy? I believe that beta cell therapy alone will not be enough, at least not in
No,
full-blown disease, and immunotherapy alone will not be enough. I
exactly.
think you're right. It could be a curative therapy if you do it in an early stage, so stage 1 diabetes, something like that.
Exactly. Exactly.
But beyond that, it should not be dismissed just because it doesn't work as a monotherapy, and it should always be done in combination with a smart immunotherapy that's non-confounding. So I think we're on the same track as always.
We have a question from Farouk Syed.
I guess main thing is we should write about this because
Yeah.
it's not well it's not well recognized in the community. Sorry.
No,
You need a
you're
synthesis
right.
piece, across.
Exactly, Monica. I agree with you.
Farouk has a question or comment.
Rene, it's amazing work. So I was wondering, have you looked into any IRE1 alpha mutation in those subjects that you guys looked in? Is there increase in insulin mRNA? Like, are they prone to develop diabetes?
With you
Because
mean
you Pfizer again knocked out IRE1 alpha in NOD mice, and she's seen some protective effect, right? So I was just wondering whether you looked into human side. Is there any relevance for IRE1 alpha mutations or any variants that can lead to onset of diabetes?
yeah, I'm not sure, to be honest. So
You know, the one thing that is odd with the IR1 alpha in mice, and it is the complete opposite, and it's not only us that see this. It's a many, and mouse insulin, either insulin 1 or 2, doesn't have the IR1 alpha motive. So whatever is happening, I think you have to be careful knocking out stuff, right? Nature is not really happy of you doing this, and it's more subtle things. So I do think that if you would interfere in the stress release pathway, be it PERK or a partial IRL1 inhibitor, you might actually have a benefit. If you do too much, it might actually go in the opposite direction. So it does indicate that you have to be careful doing this. But the fact that in mice I1 alpha interference has an effect on diabetes means that it's part of the equation. It could be in different directions in different species, but it all comes down to the same story.
Okay, but the question is, if you see CD1s, right, it's like 5 to 10 folds higher amount of insulin mRNA in their islets comparing to NODs. How does they regulate? Like basically, if they could able to, do they have like something different, IRE1 alpha mechanism in degradation of this insulin in those strains? Do you have?
You gotta ask a mouse doctor, Farouk. I mean, I don't know.
yeah, I don't know if there are SNPs in the IRE1 alpha that are linked to type 1 diabetes. Maybe in MODY, but not.
No, but that is one curious thing that I found a little surprising, together with the Exeter group, and they are world leaders in finding monogenic diseases. They carefully looked into all the SNPs that all the monogenic forms that affected ER stress, and that alone is not sufficient to cause autoimmunity. It is sufficient to cause diabetes, but it's not sufficient to cause autoimmunity. So stress is one factor, but the immune system is the other factor. And that brings me back to Matthias's point, that is actually two pathways colliding and creating the perfect storm to cause disease. But stress alone or immune response alone is not going to be the problem.
Okay, thank you.
All right, last call for questions. Okay, well, this is a great conversation.
Monica, can I just say how proud I am of Rene, my kid. Imagine that you have type 1 diabetes like Rene has, and then discover how not to get it. I mean, I think the guy rocks, and I'm very much looking forward to have him join our mission to cure type 1 diabetes.
Yeah,
This was a great presentation, Rene, indeed.
fantastic. Very
Very
good.
good, Rene.
Very well
Terrific.
done. And thank you very much for everyone who's working so hard, especially the early career scientists just jumping in. to this, all these paradigms and kind of shaking things up. So appreciate that. All right, great. Have a great rest of your day, everyone. We'll see you on the next one. Bye-bye now.
Bye-bye.
Thank you.